Hello, my name is Zoya Kadri. I'm in 7th grade from El Huda Islamic School in Hadran. My project title is The Dark Side of Sunshine, the Effect of UV Light on the Survival Rate and Behavior of Daphnia magna. Ultraviolet rays have many harmful side effects on humans, including skin cancer. In my project, I used Daphnia magna as a biosite to examine the effects of UV light on the behavior of living organisms. The purpose of my experiment is to focus on how different durations of UV exposure affect their reproduction, survival, and mortality rate. I started to research what UV light is. It is a form of electromagnetic radiation that has shorter wavelengths than visible light. The wavelengths typically range from 100 to 400 nanometers, making it invisible to the human eye. It provides vitamin D for your body, but too much exposure can greatly increase your risk of developing skin cancer. I also found out that there are many different types of UV radiation classified into three primary groups: UVA, UVB, and UVC. Each is distinguished by their wavelength. Almost all UV radiation that reaches Earth is UVA. The most common is UVA, which is less harmful to humans and is more constant throughout the year. During my research, I also found out that UV radiation alters DNA. DNA readily absorbs UV radiation, which commonly changes the molecule in one of many ways. I hypothesized that longer exposure to UV light on Daphnia would increase the mortality rate and stop cells from reproducing. I used Daphnia culture pipettes, 3 gallons of spring water, UVA lamps, white light lamp, and 9 same-size jars. I first ordered the Daphnia culture kit and cultured the Daphnia until I had enough to start the experiment. I separated the Daphnia into 3 groups: Group A, Group B, and Group C, with 20 Daphnia in each jar, and exposed each group to different durations under UV. Control Group A had no UV exposure, Experimental Group B had 24 hours of UV exposure, and Experimental Group C had 48 hours of UV exposure. The survival rate of the Daphnia was observed after 8 hours, 16 hours, 24 hours, 48 hours, and 72 hours. I observed that after 72 hours, control group A had an average 31.67 Daphnia remaining in the jar with a 58.35% reproduction rate. Experimental group B had an average 13.33 Daphnia remaining with a 33.35% mortality rate. Experimental group C had an average 10 Daphnia remaining with a 50% mortality rate. This means that they were not only dying at a rapid rate, but they also stopped reproducing. This graph shows the average Daphnia remaining in each group after 72 hours. This graph shows the population rate of the Daphnia after 72 hours. The results showed that Group A with zero exposure to UV light had the maximum reproduction rate and minimum mortality rate, while Group C had the maximum UV exposure, had maximum mortality and minimum reproduction. The results supported the hypothesis that increased UV exposure leads to increased mortality in Daphnia. Control groups showed higher survival and reproduction rate, while UV-exposed groups showed increased mortality. These findings highlight the harmful effects of prolonged UV exposure and how it has the potential to cause irreversible damage to our DNA and how detrimental the effects can be. For future research, I would like to study the effects of UVB radiation on the mortality rate of Daphnia. And that concludes my presentation. Thank you.